And the reason for that is because we're moving things against a concentration gradient, and you're going to need some type of carrier, absolutely. But that carrier is going to be using energy. It's just like you shoving all your clothes into the closet if somebody's coming over and your house is a mess. You're using energy, you're carrying that stuff to the area where it's higher concentration, and then to shut the door, it takes energy. So that's what's going to happen. But here we can see that calcium is in low concentration here inside the cell. It's higher concentration outside the cell, but through the carrier, which utilizes ATP energy, we're able to push calcium into the outside of the cell against the gradient. Whoops. Okay, so there are two types of active transport mechanisms. There's primary active transport, also known as the sodium-potassium pump. So primary active transport and the sodium-potassium pump are synonymous terms. If you ever hear primary active transport, that's what they're talking about, the sodium-potassium pump. So sodium-potassium pumps are really very important. All cells, I guess it's kind of important to note too that all these different types of transport that we're talking about, most cells are going to have maybe all forms of these transportation mechanisms. It's not like one cell just has this type or that type. All the cells have different types of transportation mechanisms that they use, and we're going to see an example of that in a few slides. But the sodium-potassium pump is very important, and all cells have it pretty much. What the sodium-potassium pump does is it uses ATP energy, and every time the pump works, it pumps 3 sodium ions out of the cell and 2 potassium ions into the cell. And what this does is it establishes a high sodium concentration outside the cell and a high potassium concentration inside the cell. And that pretty much is going to set the stage for all other processes that we have. And you're going to see how that comes into play really very soon when we get into our first body system. So anyway, remember that. Sodium-potassium pump, also known as primary active transport, uses ATP energy, and every time it uses ATP energy, it pumps 3 sodium out of the cell for every 2 potassium it brings in, making high potassium inside the cell and high sodium outside the cell. And these ions are being pumped against the gradient, which is why it requires ATP. Secondary active transport is something else. And it's very interesting, actually. It's a very interesting transport mechanism. It is moving substances against the concentration gradient. But here, the energy that's used is not really ATP. The energy that's being used is movement energy. So here's what we have. We have 2 types of transporters that can occur with secondary active transport. We can have symporters or antiporters. Now, we're going to only be talking in this class about symporters. So, you know, that's not something we're going to be really describing too much. And here they give you an example of a symporter. What's happening here is we want to move glucose into the cell, but there's already a lot of glucose in there. So how are we going to get it in there? Sodium, because the sodium-potassium pump sets things up so that there's high sodium outside the cell, high potassium inside the cell, right? The sodium that's outside the cell wants to diffuse in. It's high concentration out here, low concentration in here. So as sodium diffuses, it provides the energy to move glucose against the concentration gradient. It's a lot like drafting, I think is what it's called. You know how like people drive behind a semi-truck real close and it saves your gas mileage? Have you ever heard of that? Well, don't try it because you have to get really close and it's very dangerous from what I understand. I never did that. I actually never did that. But it's the same idea. You get energy from movement. Okay, you're getting energy from movement. In the case of the car, you're actually saving energy because you're using the energy of the truck to help pull you along. You know, that wind energy, I guess you could say. So anyhow, this is a symporter. This is secondary active transport, the sodium glucose symporter. Sodium moving with its gradient pulls glucose against the gradient. An antiporter, the molecules would move in the opposite direction. So for example, if potassium were moving out of the cell and glucose were moving in, that would be antiport. Okay, so on the previous slide, it actually explains it. I think this is a pretty decent explanation. The energy created by sodium moving with its gradient brings another molecule, in the case we just saw, glucose, into the cell against its gradient. So that's symporter. And that's secondary active transport. Any questions? All right, now we're going to put it all together. As I mentioned before, most cells do in fact have multiple transportation mechanisms. It's not like they just have a sodium-potassium pump or they just have facilitated diffusion. They have multiple transporters in order to perform different functions. So this diagram, I would put a star beside this because this is a good place where you can study. You can study this, you can talk about it, and you'll actually be studying the different transport mechanisms, with the exception, I think, of like osmosis and simple diffusion. Pretty much you'll be able to study your transportation mechanism in a functional way. So there are two ways that we can bring things into the bloodstream. There's absorption, which occurs in the digestive tract, and that's where we bring digestive products across the intestinal epithelium into the blood. So that's absorption of nutrients. Then we have reabsorption, which occurs in the kidneys. This is where we move substances out of the filtrate that the glomerulus and Bowman's capsule, you know, you filter stuff out of the blood, you move it back into the blood. So we call that reabsorption because we absorbed it before, it was already absorbed, but we filtered it out and now we're reabsorbing it. The digestive tract, of course, those nutrients haven't been absorbed. They are, you know, just there and you bring them in for the first time into the bloodstream. Okay, so this is, we're going to pretend a kidney cell, and you can see these are probably simple cuboidal and you have little microvilli. So this is probably the proximal convoluted tubule where a lot of reabsorption takes place of water, glucose, stuff like that. And this down here would be where your bloodstream is. So our goal in this diagram is to go here from the lumen of the kidney tubule, probably the proximal convoluted tubule, to get glucose that's in here in the filtrate back into the bloodstream. That's what we want to do. So we do this through these different transport mechanisms. So first, we have primary active transport, also known as the sodium-potassium pump. And what happens is, again, ATP is utilized to pump 3 sodiums out for every 2 potassiums it brings into the cell. Now, keep in mind, they're only, for the sake of simplicity, they're only drawing this sodium-potassium pump here. But you would also have sodium-potassium pumps here as well. And so ultimately we're going to have high sodium concentration outside the cell and high potassium concentration inside the cell, which is set up by the primary active transporter, the sodium-potassium pump. Then the ion gradients of sodium and potassium will set the stage for secondary active transport to occur here. Okay, now secondary active transport, we just talked about the sodium glucose symporter. So because we have high sodium outside the cell, because of the sodium potassium pump, Sodium will diffuse into the cell following its gradient to pull glucose into the cell against its gradient. Glucose then, being in high concentration inside the cell and lower in the blood, through facilitated diffusion, is going to be able to move passively into the bloodstream. So it goes from high concentration to low, but it is too large, so it requires a carrier protein, which again is facilitated diffusion. And then ultimately the glucose is brought into the bloodstream, and that's reabsorption. That's how that occurs. So let's talk about it once more. First, we have the sodium-potassium pump, primary active transport, on both sides of the cell, pumping 3 sodium out of the cell for every 2 potassium it brings in, using ATP energy. So sodium's in high concentration, excuse me, outside the cell. So sodium can diffuse into the cell with the secondary active transport. Excuse me. Uh-oh. Now I'm going to have a bunch of sneezes and coughs. So sodium, through the secondary active transport, sodium will move with its gradient, pulling glucose against its gradient into the cell. Now glucose is in high concentration in the cell, low concentration in the blood, so facilitated diffusion, which just requires the concentration gradient and that transporter, will move it from high concentration in the cell to low concentration in the blood. Oh, yeah. Okay, I think I'm better now. Any questions about that? Does that help a little bit to tie things together for you somewhat? A little bit? No questions at all? This is a very important diagram. You should definitely study this. Okay, so transportation across membranes, the epithelial membrane specifically, can occur in one of 2 ways. One, transcellular transport, which is what we just described. We move material from one side to the other of the epithelial cell, going through the cytoplasm of the cell. That's basically what we just described. Paracellular transport moves material through tiny spaces between the epithelial cells. And that can be a little tricky because there are a lot of junctions in between epithelial cells that will prevent this type of movement. So there are complexes of junctions, as we talked about, that connect adjacent cells. And generally, epithelial cells will have the different ones, not just, one or the other. Generally, the apical surface, you're going to have the tight junction. This is so things cannot get in between the cells from the lumen, like the digestive system, for example. You don't want to have like nutrients passing in between the cells and the bloodstream. That wouldn't be very highly regulated. So the tight junctions help to prevent that. We also have adherence junctions. This is where we've got the membranes, they're basically glued together and they're anchored in place by these cytoplasmic proteins. And then we have the desmosome junctions, which have these little protein buttons that help to anchor these little fibers, which kind of stretch a little. They're more elastic. They can be pulled apart. So you'll find a lot of desmosome junctions in between, for example, cardiac muscle cells. I mean, we're talking about epithelial tissues here, but cardiac muscle cells would also, because they have to kind of stretch. When blood fills the chambers of the heart, they'll stretch a little bit. So anyway, those are the different types of junctions. I know you talked about them in anatomy, but over here you can see an electron micrograph, which does show that, again, in between certain epithelial cells, you will have multiple types of junctions that are found between them. And then, of course, the plasma membrane also can move large molecules by way of endocytosis and exocytosis, which we already talked about previously. But of course, that's a very different type of transport than those transport mechanisms we talked about today. So, any questions? Okay, I'm always worried when you don't have questions. All right, so the next thing we're going to describe is probably the most, I think, complex of all the topics in this particular lecture chapter. This is probably the most complex. This chapter is like mental calisthenics. It's like interval training. It's like it's easy, then it gets harder, then it's a little easy, then it gets really hard, and then it's like it ends up kind of easy, like neutral. So this topic is one that I usually like to cover at the beginning of class when people are the most fresh. But unfortunately, we're getting into towards the end of the class. But this is it's important because this topic is all about pretty much how the inside of the cell has a particular charge relative to the outside of the cell. How is it established? Why is a charge separation so important? Because any cell in the body that can do something, like for example, a neuron can transmit electrical signals. It has to have a charge inside the cell when it's resting that can be disrupted to ultimately get it going to do what it is it's supposed to do. Okay, and so this little section of chapter 6 is about how we establish that charge inside the cell relative to the outside. And then the rest of the course, when we talk about the systems, is going to be about how we disrupt that and how things happen ultimately. So this is membrane potential. Membrane potential specifically describes the difference in charge across membranes. So we actually measure the electrical charge inside of the cell in very small measures of voltage called millivolts, because it's very, very small. So we have, again, for example, cardiac muscle cells, the membrane potential when the cell is at rest, not contracting, is minus 90 millivolts. That means that its charge is less inside the cell than the outside the cell, than it is outside the cell. So by 90 millivolts. So that's what negative 90 millivolts means. It means it's minus 90 millivolts inside the cell relative to the outside. Nervous tissue, neurons have a resting membrane potential of minus 70 millivolts. Okay, so how do we establish this? Okay, before we go through these notes, I'm going to, I think I'm going to go ahead and just draw this out for you, because personally, I think it's, like I said, if you go into these notes, I think without really having sort of an overview, it makes it a little bit trickier. Personally, I think that. So I'm going to turn this off for a second. Can I turn on the light? I know we've been sitting in the dark all day, but I can turn on the light. I mean, everybody chose the lights to be off. I tried to acquiesce. Okay, I'll tell you what. I'll compromise. I'll just put them on in the front. Let's see. Okay, and you guys are still in darkness. All right, so there are three major variables that contribute to resting membrane potential. Okay, so we have, first, we have the sodium-potassium pump. this is our cell. And throw our little sodium potassium pump in here. So remember, we use ATP to pump what? ATP to do what? What's the sodium potassium pump do?
Free sodium out.
It's in the name, right? It pumps sodium and potassium. Right, three sodiums out for every 2 potassiums it brings in. Okay, so 2 potassiums go in. You guys are killing me. 3 sodiums out. Okay, so 3 sodiums out for every 2 potassium it brings in. All right, so that's the first thing, sodium potassium pump. It establishes high sodium inside, high sodium outside, high potassium inside. Okay, the second thing is that we have proteins which have a negative charge. Okay, so negatively charged proteins will attract positively charged ions. And the third thing is that membrane is more leaky to potassium than it is to sodium. Okay. So what direction would, so again, we talked about diffusion. We talked about how sometimes we need to have these diffusion, these channels. In this case, for charged particles and so forth, right? In this case, we have diffusion channels for sodium and for potassium, okay? And they're always open. What direction will sodium diffuse, in or out of the cell? Where is sodium concentration highest? Outside, right. It's highest outside, so it's going to diffuse where? Inside, right. So sodium will diffuse in. Potassium then will move where? Where's potassium highest? Inside, so it's going to move out, right. So potassium will diffuse out. Okay, so these are 3 variables, okay. So first of all, our sodium potassium pump establishes the high sodium outside, high potassium inside the cell. Okay, now the fact that we have these leakage channels, if the membrane were only leaky to potassium, we would lose a lot of positive charges, right? At the point at which we would have 1 potassium leaving for every 1 potassium that came in because of these negatively charged proteins that are here, the resting membrane potential would be very, very low. I mean, it would be like -90. I mean, it would be very negative because we lose so much of the positive charges. If the membrane were only leaky to sodium, our membrane potential would be a lot more positive because we would have a lot of positive charges coming in. But because of the fact that we have both sodium and potassium that can diffuse, but the membrane loses more positive charges through the potassium and we gain positive charges through the sodium, our resting membrane potential is going to not be so positive, it's not going to be so negative, it's going to be in the middle at about -70 on average. So that's what the rest of the notes is going to tell us about, is basically what we've drawn up here. So I'm going to go through the notes, but I want to repeat this one more time so that you understand. So again, the negative charge inside the cell versus outside the cell, which is known as the resting membrane potential, is established by three things. First, the sodium-potassium pump that establishes high sodium outside, high potassium inside the cell. Secondly, these negatively charged proteins. And third, the fact that we have more potassium leakage channels than we have sodium. So essentially what's happening is we are losing more positive potassium charges than we're gaining sodium coming in. Negatively charged proteins help to draw that in. And because of that, our resting membrane potential isn't super positive, like if we were just having sodium come into the cell, or super negative, like if we were having potassium just leave the cell. It's going to be in the middle at about -70 millivolts. And that resting membrane potential literally is that charge inside the cell when the cell is not doing anything. And in physiology, that membrane potential will be disrupted when certain things have to happen. Okay, like a neuron has to transmit a signal, or a muscle has to contract, something like that. Okay, so let's go through the notes. And again, we'll just repeat what it was that I just told you, only the slides are going to be a lot busier. And they can look a lot more confusing than what they are. Oh, no, hang on. This is why I don't like to turn it off, but you don't really have a choice. I always wish that they would have this screen be a little smaller so you have more board, you know, then you don't have to turn everything off. Okay, so this is, again, the first slide. So the membrane potential is the difference in charge across the membranes. More negative inside the cell than outside. It results in part from the presence of large anions, which are these proteins that are trapped inside the cell. They're too big, they can't get out. Diffusible cations, such as potassium, are attracted into the cell by these anions, the negatively charged proteins. Sodium is not as permeable, though, as we said. So these are some of the things that establish that. Now, what this is explaining, this equilibrium potential, is what I explained to you earlier, which is if the membrane was only permeable to potassium, essentially it would diffuse out of the cell, going with its gradient. And it would continue to do that until it reached its equilibrium potential, which is basically the point at which you're losing 1 potassium for every 1 potassium that is attracted into the cell by the anions, okay? At that point where we reached equilibrium, you would have a charge inside the cell of about -90 millivolts because we're losing more positive charges than we're gaining. So it's going to be very negative. But if the membrane was only permeable to sodium, that means that the sodium would be coming into the cell, right? And at the point where it reached equilibrium, where you have 1 sodium coming in for every 1 sodium that was leaving, because we're gaining more positive charges, we would have a very positive resting membrane potential of about +66. But because we have both ions, but the membrane is more leaky to potassium than to sodium, so we're actually losing more positive charges and we're gaining, we end up with a negative resting membrane potential around minus 70 millivolts. So the resting membrane potential is the membrane voltage of a cell not producing impulses, as I said, at rest. If your neurons are just sitting there and you're not using them, or if your heart isn't beating or your skeletal muscles are resting, you're not contracting them, this is what the charge would be. The resting membrane potential of most cells is between minus 65 and minus 85. Again, sometimes, you know, like with cardiac muscle tissue, it's about minus 90 millivolts. Neurons are about minus 70 millivolts. The resting membrane potential depends on the concentrations of ions that are inside the cell and out, and the permeability of each ion. And as I said, because the membrane is more leaky to potassium, it plays the larger role. It has a bigger influence. So again, we have potassium diffusing out, sodium coming in. Not as much sodium is coming in as potassium is leaving. So because of this, the resting membrane potential is less negative than if it were just potassium alone that we're moving, right? Because we're adding some positive charges back in with the sodium. So it's not going to be super duper negative. And then we have the sodium potassium pump, which also plays a role. This diagram at the very end shows us the three variables that I was talking about on the board, but the processes that affect the resting membrane potential help to establish the resting membrane potential: the negatively charged proteins, the fixed anions, the unequal permeability of potassium and sodium, the membrane's more permeable to potassium and less permeable to sodium, and the sodium-potassium pump establishing higher potassium inside and higher sodium outside the cell. All of these variables ultimately will get us to the point where the resting membrane potential will average out to be about minus 70 millivolts for most cells. And if you want to know math and you want to do the mathematical equation, in the textbook there is a whole section on the Nernst equation that you can go through and you can actually calculate how you come to this point. Because mathematically it doesn't work out. If you look at the equilibrium potential values, it doesn't work out to minus 70. It's a more complicated equation. But it's really, I think, beyond the scope of what we need to know here. Any questions? So what I drew on the board, those are the three major contributions or contributors to the resting membrane potential. Okay, so we have about 4 minutes. This last part of chapter 3, or 6, sorry, we're in chapter 6, is really not so complicated, which is nice. Although I don't know how you feel at the end of the class, it may seem more complicated than what it is. Cell signaling. Cell signaling is basically how cells communicate with one another. We can communicate with each other through cell phones, through a cell phone tower, something like that. We can communicate by text, we can communicate by email, we can communicate by, you know, whatever app you use. We can communicate in many different ways, and cells do that as well. But usually cells are going to use things like ions, or they're going to use things like neurotransmitters, which are chemical signals. Some cells use gap junctions through which signals pass directly from one cell to the next, like cardiac muscle tissue, gap junctions at the intercalated discs. There, we have ions that flow from cell to cell. Sometimes we use a chemical signal, and a target cell has to have a receptor protein for a chemical signal. There are different types of chemical signaling. in the endocrine system, we have paracrine signaling, which is where cells secrete regulatory molecules that are going to travel a short distance to a target cell. And an example of this would be what happens in the digestive tract. Whenever you eat food, then basically the stomach is going to release CCK or cholecystokinin, which is going to act on the small intestine and the pancreas so that you can get more secretion, so that the area of the small intestine, the duodenum, is going to be ready for that food, that chyme, that real acidic chyme to come into that area. So that's an example of paracrine signaling, where you just release a signal, it travels a very short distance to a target tissue. Let me skip that for a second. Endocrine signaling is where cells secrete a chemical that moves through the bloodstream to distant target cells. An example we talked about today, we were talking about regulation of blood osmolality, right? We said that the posterior pituitary gland in the brain is going to travel, the signal that it releases, the antidiuretic hormone, is going to travel all the way down to the kidneys. That's an example of endocrine signaling that occurs. And then the previous slide, we have synaptic signaling, which happens between neurons. And this specifically, the type of message that we have is a neurotransmitter. So we can use hormones like in paracrine or endocrine signaling. We can use synaptic signaling where we're using neurotransmitters. There's different types of messages that can be sent. Or we can use ions, like if we're talking about electrical signaling. Okay, so I think we can stop here for today. And what do you say? You think we have a quiz maybe on Tuesday? We had talked about that earlier, but I don't know, because I'm just concerned. I want to make sure you guys have had some questions for this material for this week, and since we don't have class on Thursday, we're not going to be able to do that. So what do you think? You want a quiz Tuesday? I mean, you don't want it, but I mean, you know, it's sort of like a shot. Nobody wants a shot, but you know it's good for you. Or, I mean, like a shot, like, wow. It's going to be like a...
That would like more than one shot. So.
I wasn't thinking bonus, but I don't know. Maybe we could work it out somehow. I'll have some bonus question on there or something. I don't know. Yeah, let's do that. Let's have a quiz Tuesday on the stuff from this week. So it'll cover everything from proteins, because we didn't have any questions on proteins, nucleic acids, and then to this slide, which is slide 71 in the chapter 3 and 6 lecture. Okay.